The vascular system in plants. The vascular system in plants is a specialized network of tissues that transport water, nutrients, and other substances throughout the plant body. It consists of two types of tissues, xylem and phloem. The xylem transports water and minerals from the roots to the shoots, while the phloem transports sugars and other organic compounds from the sites of photosynthesis to the rest of the plant. The vascular system plays a crucial role in the growth and development of plants, providing structural support and enabling the distribution of resources necessary for survival. The roots. Roots are plant organs that serve several functions, including anchoring the plant in the ground, absorbing water and nutrients from the soil, and storing food reserves. Roots can be classified into two main types, taproots and fibrous roots. Taproots are thick, elongated roots that grow deep into the soil, while fibrous roots are network-like and spread out horizontally close to the surface. Both types of roots have root hairs, which increase the surface area of the root and facilitate water and nutrient uptake. Additionally, some roots can form symbiotic relationships with fungi or bacteria, which can help the plant acquire nutrients from the soil. Overall, roots are essential for plant growth and survival, and their structure and functioning can vary depending on the plant species and environmental conditions. Cross-section of a dicot root. A cross-section of a dicot root typically shows the following structures, from the outside towards the center. 1. Root cap, the tip of the root that protects its growing tip. 2. Epidermis, a single layer of cells covering the root surface that helps in absorption. 3. Cortex, a layer of parenchyma cells that stores food and helps in radial movement of water and nutrients. 4. Endodermis, a single layer of specialized cells that surrounds the vascular tissue and controls the entry of water and minerals into it. 5. Pericycle, a thin layer of cells that gives rise to lateral roots. 6. Vascular tissue, consists of two types of tissues, xylem and phloem. Xylem tissue helps in water conduction and phloem tissue helps in the conduction of organic molecules. 7. Pith, a central region composed of parenchyma cells, which stores food and supports the stem. Note that the arrangement and composition of these layers may vary depending on the plant species and age. Fig. 4.23, cross-section of a dicot root. Cross-section of a monocot root. A cross-section of a monocot root typically shows the following structures, from the outside towards the center. 1. Root cap, the tip of the root that protects its growing tip. 2. Epidermis, a single layer of cells covering the root surface that helps in absorption. 3. Cortex, a layer of parenchyma cells that stores food and helps in radial movement of water and nutrients. 4. Endodermis, a single layer of specialized cells that surrounds the vascular tissue and controls the entry of water and minerals into it. 5. Vascular tissue, consists of 2 to several layers of small bundles of xylem and phloem, distributed around the circumference of the root. 6. Pith, a central region composed of parenchyma cells, which stores food and supports the stem. Note that monocot roots have no distinct pericycle, and the arrangement and composition of these layers may vary depending on the plant species and age. Fig. 4.24, cross-section of a monocot root. The stem. The stem is one of the main organs of the plant body that supports leaves, flowers, and fruits, and conducts water and nutrients throughout the plant. It consists of different types of tissues, including epidermis, cortex, vascular bundles, and pith. The epidermis is the outermost layer of cells that covers the stem surface and helps to protect it from water loss and physical damage. The cortex is the layer of cells between the epidermis and the vascular bundles that stores food and provides mechanical support to the plant. The vascular bundles, which are composed of xylem and phloem tissues, are responsible for transporting water, minerals, and organic compounds throughout the plant. The xylem transports water and minerals from the roots to the rest of the plant, while the phloem transports sugars and other organic molecules from the leaves to the rest of the plant. Finally, the pith is a region composed of parenchyma cells located in the center of the stem that stores food and provides structural support. The anatomy of the stem can vary depending on the plant species, its age, and growth conditions. Cross-section of a monocot stem. A cross-section of a monocot stem typically shows the following structures, from the outside towards the center. 1. Epidermis, the outermost layer of cells that protects the stem and helps to prevent water loss. 2. Cortex, a layer of parenchyma cells that stores food and provides mechanical support to the stem. 3. Vascular bundles, scattered throughout the stem, usually in a circular pattern. Each vascular bundle consists of both xylem and phloem tissues, with the xylem tissue located towards the inside and the phloem towards the outside of the bundle. 4. Pith, the central region of the stem composed of parenchyma cells that store food and provide structural support. Note that monocot stems do not have distinct secondary growth, and the arrangement and composition of these layers may vary depending on the plant species and age. Fig. 4.25, cross-section of a monocot stem. Cross-section of a dicot stem. A cross-section of a dicot stem typically shows the following structures, from the outside towards the center. 1. Epidermis, the outermost layer of cells that protects the stem and helps to prevent water loss. 2. Cortex, a layer of parenchyma cells that stores food and provides mechanical support to the stem. 3. Vascular bundles, arranged in a circular pattern surrounding the pith, with larger bundles usually located towards the outside of the stem. Each vascular bundle contains both xylem tissue, towards the inside of the bundle, and phloem tissue, towards the outside of the bundle. 4. Cambium, a thin layer of meristematic cells between the xylem and phloem tissues that can give rise to both secondary xylem, wood, and secondary phloem. 5. Pith, the central region of the stem composed of parenchyma cells that store food and provide structural support. Note that dicot stems typically have the ability to undergo secondary growth, resulting in the formation of wood. The arrangement and composition of these layers may vary depending on the plant species and age. Components of the vascular system. The vascular system is composed of two main types of tissues, xylem and phloem, that are responsible for transporting water, minerals, and nutrients throughout the plant. 1. Xylem, this tissue is responsible for transporting water and minerals from the roots to the rest of the plant. It is made up of several types of cells, including tracheids, vessel elements, fibers and parenchyma cells, which are arranged in different patterns depending on the plant group. Tracheids and vessel elements are specialized cells that make up the water-conducting channels, while fibers provide mechanical support, and parenchyma cells store food. Xylem is made up of xylem vessels and tracheids. Xylem vessels are specialized cells made of vessel elements that are responsible for transporting water and minerals from roots to the upper parts of the plant. The vessel elements have perforated end walls that allow water to move efficiently from one cell to another, forming a continuous tube without interruption. Tracheids are long narrow cells that make up the majority of the xylem tissue in most vascular plants. They are tapered at each end, and their walls contain pits, which are regions where the secondary wall is absent or thinner, allowing water and minerals to move easily between adjacent cells. Unlike xylem vessels, tracheids are not specialized to transport large volumes of water quickly. However, they provide structural support and help to maintain the structural integrity of the xylem tissue. 2. Phloem, this tissue is responsible for transporting sugars and other organic molecules from the leaves to the rest of the plant. It is also composed of different types of cells, including sieve elements, companion cells, fibers and parenchyma cells. Sieve elements are specialized cells that form the sugar-conducting channels along with the companion cells, which are responsible for metabolic support of the sieve elements. Fibers provide mechanical support, and parenchyma cells store food. Phloem is made up of sieve tubes and companion cells. Sieve tubes are specialized cells in the phloem tissue that are responsible for transporting sugars and other organic molecules throughout the plant. They are elongated, tube-like cells that lack nuclei, ribosomes, and vacuoles, and their cytoplasm is confined to a thin layer near the cell membrane. The sieve tube cells are connected end-to-end via sieve plates, which have pores that allow sap to move from one cell to the next. Companion cells are specialized cells that are closely associated with sieve tube elements in the phloem tissue. They are responsible for providing metabolic support to sieve tube elements by synthesizing and transporting proteins, RNAs, and other molecules into the sieve tube element cytoplasm. Companion cells contain nuclei, ribosomes, and mitochondria, and they are connected to the sieve tube elements by plasmodesmata, which allow direct communication between the two cell types. Xylem and phloem tissues are usually located in close association with each other, forming vascular bundles that run throughout the plant body. The arrangement and structure of these tissues and bundles will vary depending on the plant species, age, and the organ that they form part of. Fig. 4.27, longitudinal section of a stem. Longitudinal section of a stem. A longitudinal section of a stem is a cross-section taken along the longitudinal axis of the stem, resulting in a view that shows the internal structure of the stem. The following structures would be visible in a longitudinal section of a stem. 1. Epidermis, a layer of cells that covers the outer surface of the stem, providing protection and absorption. 2. Cortex, a layer of cells located beneath the epidermis that stores starch and other nutrients. 3. Vascular bundle, a group of xylem and phloem tissues that run through the center of the stem. The xylem is usually located towards the inside of the stem, while the phloem is located towards the outside. 4. Pith, a central area of tissue that is surrounded by the vascular bundle, composed of parenchyma cells that store food. 5. Cambium, a layer of cells located between the xylem and phloem that can differentiate into new cells in both directions, allowing the stem to grow in diameter. The exact arrangement and distribution of these tissues will depend on the plant species and the age of the stem. Fig. 4.28. A. Xylem vessels. B. Tracheids. Revision exercise 4.8. 1. Explain the difference between xylem and phloem. Xylem and phloem are two types of vascular tissues found in plants. The main difference between them is their function. 1. Xylem tissue, responsible for transporting water and minerals from the roots to the other parts of the plant. Xylem is made up of tube-like structures called vessel elements, and long, tapered structures called tracheids. These structures have thick secondary cell walls reinforced with lignin, providing rigidity and support to the plant. 2. Phloem tissue, responsible for transporting food, sugars, amino acids, and other organic nutrients, from the leaves to the other parts of the plant, including the roots. Phloem is made up of two types of specialized cells, sieve tube elements and companion cells. The sieve tube elements form long tubes that are connected by sieve plates, which allow the movement of nutrients. Companion cells provide metabolic support to sieve tube elements. In summary, xylem tissue transports water and minerals, while phloem tissue transports food and plants. 2. Give a brief definition of vascular bundles. Vascular bundles are bundles of xylem, phloem, and other specialized cells found in various parts of plants, including stems, leaves, and roots. Vascular bundles provide structural support to the plant and are involved in the transport of water, nutrients, and other substances throughout the plant. In stems, vascular bundles are usually arranged in a ring shape, with the xylem located towards the center and the phloem towards the outer edge. In leaves, vascular bundles are arranged in a branching pattern, with the main vein running through the center of the leaf and smaller veins branching off from it. The exact arrangement and distribution of vascular bundles can vary depending on the plant species and the specific organ being examined. 3. Distinguish between monocot roots and dicot roots using the arrangement of vascular bundles. Monocot roots and dicot roots can be distinguished based on the arrangement of their vascular bundles, as follows. 1. Monocot roots typically have a circular arrangement of vascular bundles, with the xylem and phloem tissues arranged alternately in a ring around the center of the root. In monocots, the number of vascular bundles is usually several, such as 6 or more, and distributed evenly around the root. 2. Dicot roots typically have a centralized arrangement of vascular bundles, where the xylem tissues are found towards the center of the root, arranged in the shape of an X or H, and the phloem tissue located on 4 sides of the xylem. In dicots, the number of vascular bundles is usually 2 to 4, and arranged centrally in the root. The arrangement of vascular bundles is an important characteristic used to differentiate monocots and dicots, particularly in the root structure, but also throughout other parts of the plant. 4. State the difference between xylem vessels and tracheids. Xylem vessels and tracheids are two types of specialized cells found in xylem tissue, but they differ in structure and function as follows. 1. Xylem vessels are hollow, tube-like structures in the xylem tissue that are responsible for the transport of water and minerals from the roots to the other parts of the plant. Xylem vessels are composed of dead cells that are joined end-to-end, forming a continuous column for water movement. The walls of xylem vessels are perforated with small pores called pits, which allow water to move between adjacent vessels. 2. Tracheids are elongated cells found in xylem tissue that are also responsible for water transport. Unlike xylem vessels, tracheids are long, narrow, and tapered, with thick secondary cell walls reinforced with lignin. Tracheids have pores called bordered pits, which allow water to move laterally between adjacent cells. In summary, while both xylem vessels and tracheids are involved in water transport in plants, xylem vessels are wider, shorter, and more efficient at water movement, while tracheids are elongated, narrow, and more resistant to collapse under tension. 5. List any 2 adaptations of root hairs to function. Root hairs are small, elongated, finger-like projections that grow from the surface of plant root cells and function to increase the surface area of the root for absorption. Here are 2 adaptations of root hairs to function. 1. Large surface area. Root hairs have a large surface area to volume ratio, which allows them to absorb water and nutrients efficiently. Their elongated shape maximizes surface area by extension, increasing the area over which water and nutrients can be absorbed. 2. High concentration of nutrient transporters. Root hairs have a high concentration of nutrient transporters, such as aquaporins and ion channels, embedded in their plasma membranes. These transporters facilitate the movement of water and nutrients into the root hairs and across the root epidermis, which speeds up nutrient uptake by the plant.